Hello, my name is Barbara Delgado. Welcome to my presentation this week's module reading, Label-Free Microarray Imaging for Direct Detection of DNA Hybridization and Single Nucleotide Mismatches, journal article from Boston University. In this presentation, I will be discussing the terminology, traditional methods of fluorescent labeling of microarrays, the problem with that, and the proposed solution from this research. In this particular experiment, DNA hybridization is performed with fluorescent labeling. DNA hybridization is a technique that measures the degree of genetic similarity between pools of DNA sequences. The method of hybridization is first the DNA of one organism is labeled. Then it is mixed with unlabeled DNA to be compared against. The mixture is allowed to mix and create renewed hybrid double-strand DNA. The hybrid DNA is carefully melted, and the reaction of each piece melting has information regarding the similarity of the two organisms. As you can see on the right image, pictorially describes what hybridization actually is. The labeling used for this DNA hybridization is fluorescent labeling. Fluorescent labeling is attaching a biomolecule with another molecule that stains or chemically labels the molecule of interest. These labeled molecules will emit different types of light when analyzed. These light-emitting DNA sequences are placed in a microwave for further analysis. An example is shown on the bottom image. A microarray is a laboratory tool used to detect the expression of thousands of genes at the same time using microscope slides that are printed in an array with each element being a known DNA sequence or gene. As described in the previous slide, traditional fluorescent-based detection methods involve using fluorescent intensity seen in the microarray to quantify the genetic expression levels. In order to make this repeatable, the probe must maintain uniform and consistent coverage across the whole microarray. This is extremely difficult and involves complex procedures. It also has not been very reliable in the reproducibility of the microarray data and is extremely difficult, leading to questionable results most of the time. In this research, optical interferometry uses optical interferometers, a device in which two or more light waves are combined together to produce interference. This is typically used in astronomy. This research uses a spectral reflectance imaging biosensor, or SRIB. The SRIB measures the change in optical thickness of a biolayer as a result of mass accumulation and provides quantitative information about the interactions between the analytes and the immobilized probes. This journal article describes the successful implementation of this label-free microarray imaging for direct detection of DNA hybridization and single nucleotide mismatches. This biosensor can detect the hybridization of DNA and also identify single mismatch duplexes by studying the denaturalization kinetics. It is a straightforward and accurate method for high throughput sensing for DNA analysis. So what's next? There is great potential in this new method of label-free microarray imaging. It can be implemented in numerous fields of medical study like cancer research, genealogy, and pharmaceuticals. It seems that numerous researchers have progressed with this research further and hopefully will have a common method in medical. Increasing the accuracy and results will greatly help the diagnosis of cancer and other dangerous diseases that are related to genetics. That is the end of my presentation, and I look forward to reading your comments and questions. Have a great week.